If you live in UK, you know that there is this whole fiasco about COVID-19 testing, website not working, not enough tests being done. There is a lot of things going on. Hey, it's me, Dr. T, and today I want to talk to you about COVID-19 testing. So why do you do testing? You do testing in order to know whether a person has the virus or doesn't have the virus. And also on the population level or the national level, it's good for governments to see how good they are at controlling the spread of the virus, the extent to which the virus is affecting society. The main test, or the gold standard test that is being used for COVID-19 testing, is known as qRT-PCR or RT-PCR. That is a mouthful, but the whole name is actually a bigger mouthful. So it's quantitative reverse transcription polymerase chain reaction. So RT-PCR, qRT-PCR, this is something that is used in biological science research a lot. The whole idea behind RT-PCR, qRT-PCR is to kind of see whether a particular gene is expressed or how much it is expressed. If you don't know what a gene is, I made a separate video about that, which I will link in the description box and also at the end of the video. So the principle behind it, or the protocol that is done for this RT-qPCR is that firstly you'll do a swab. The swab will be in the nose throat area, so it's a nasopharyngeal swab, and it's done in this nose throat area because COVID-19 is a respiratory disease. You're more likely to have more virus in those areas. So you do a swab, and the swab, if it has the virus, you don't want the person who is doing the test to be infected, so you put the swab in some sort of solution which will inactivate the virus. And so the virus has genetic material known as RNA, so that RNA has the information to make more viruses. And you also, as a human being, you also have your own RNA. Anything else that's living in your throat, maybe the good bacteria that lives in your throat or nose area, anything else that's living there will also have RNA. For it to be used in the polymerase chain reaction, needs to be converted into DNA. So we call this reverse transcription. So the RNA being converted into DNA. So remember, you're converting all the RNA that you have into DNA. Your RNA, viral RNA, bacterial RNA, anything RNA is being converted into DNA. Now you have your DNA, it's time to perform the PCR or the qPCR, the quantitative PCR. So in order to just isolate any material, any genetic material from the virus, the DNA that corresponds to the virus, we have specific molecules known as primers, which will help to isolate material from the virus. So we have all of this stuff in reaction mixture. You have the DNA, you have the primers, and you have some other stuff, and you perform the polymerase chain reaction. So it's a machine that does the polymerase chain reaction. This machine is called a thermocycler. It's called a thermocycler because the PCR is performed in cycles, and also because the temperature changes within each cycle. So each cycle, the temperature is changing several times. Reaction will just amplify the DNA from the virus. In order to detect the amplification of the DNA, the reaction mixture that you've put into the machine will have some sort of fluorescent dye or a probe that will fluoresce in the presence of double-stranded DNA. So the more double-stranded DNA is produced, the more fluorescence you get. This part now is more important for you to understand and listen to, so make sure your thinking caps are on. We're looking at fluorescence in order to determine how much DNA is being amplified, how much new DNA is being produced. Okay, so naturally, or at the baseline level, there is background fluorescence. So imagine this background fluorescence is represented by a pen here. And then with each cycle, you're producing more DNA, so more fluorescence. So imagine this is your cycles, 1 cycle, 2 cycle, 3 cycle, etc., etc. Eventually, you'll reach a cycle in which the fluorescence is more prominent or more detectable or surpasses the background fluorescence. So this cycle here is known as your cycle threshold. So if you already have loads of DNA from the virus to begin with, imagine this, you only need fewer cycles in order to surpass the background fluorescence. But if you have less virus to begin with, you need far more cycles in order to surpass the background fluorescence. The cycle threshold, if you had less virus to begin with, is much higher than a cycle threshold if you had more virus to begin with. Of course, if you have no virus, there's nothing to amplify, so usually no number will be given, no cycle threshold will be given. So this is the gold standard test. It's really great because it's sensitive, so even if there's a small amount of virus, it can detect it. The only thing is that sometimes it can be too sensitive. And so supposing you have very little amounts of virus there, you're very unlikely to be spreading it, or you may have cleared the infection, maybe just have a bit of genetic material from the virus just floating about. The PCR test, this RT-qPCR, qRT-PCR, is going to detect it. And if the person or whoever is giving the results doesn't give doesn't say positive or negative, we could detect it, we couldn't detect it, and doesn't give the cycle threshold, you don't know the extent to which the person is infectious. So it's really important, a lot of scientists are saying this, that they report the CT values, the cycle threshold values, alongside the positive or negative. So another downside about PCR is that it's very expensive. So first of all, you need the people doing it to be trained. So you have to have some level of expertise. Not everyone can do it. Second of all, the reagents that you use are also quite expensive. Third of all, the machine you use is quite expensive. It's a Thermocycler machine because it's going through different temperatures. You can't just use any old machine. And this machine, I'm not talking about hundreds of pounds. It can be thousands, tens of thousands of pounds. And also it takes a long time to do. The actual PCR part is not like minutes. We're talking hours. So maybe 2 hours, maybe 3 hours, 4 hours. So it's not a quick thing. Some companies have used this technology and they've been able to kind of optimize it and make it shorter and make it so you need less expertise to do it. So I came across a company known as DNA Nudge, which already have their own sort of technology, which have developed COVID Nudge. So basically you just take a swab, you put it in a cartridge, this cartridge is put in their special box, their special machine, and then it does the RT-PCR on the machine. I don't know how specifically, I don't know what step specifically, but then this information, whether it's like positive, negative, inconclusive, failed, this information is then uploaded to the cloud. Then the clinician doing the test has an app, and then the clinician can tell you whether you're positive, negative, that kind of thing. So DNA Nudge is cool that you can get your results within an hour. I don't know what level of optimization that they've done, but it sounds like a really cool technology if you can get your results within 1 hour. I also want to mention that I'm not sponsored by any of these companies. No one is paying me to say this. It's just from what I've read. So PCR is quite expensive. It takes a long time to do. And I came across this other technology known as RT lamp, which the first time I heard about this was when I was reading about these COVID-19 testing. RT lamp stands for loop mediated isothermal amplification. We'll just say lamp. Kind of like the qPCR, the principle is just to isolate the RNA, convert it to DNA, and then amplify the viral genetic material to kind of say whether you're positive or negative. That's the kind of idea, whether the viral material could be detected, viral material couldn't be detected. The thing about lamp is that generally you only need one temperature, so you don't need that expensive PCR machine, which is the Thermocycler machine, and also the reagents cheaper than PCR, and also it amplifies genetic material quicker, so generally it doesn't take as long as a PCR. This one, depending on the specific protocol or the specific reagents they use, you can look at, it will be a colorimetric test, so it will change color in the presence of the virus, so the machine will detect that, or you can like visually look at it and be like, okay, well, this sample is positive or this sample is negative. Some of them, they've optimized it as well that you can just use saliva, which is also really cool, so you don't need to use the swab. So there are some kits or some tests that if you have strong positives, it can tell you within 20 minutes without having to extract the RNA. But if you don't have a strong positive, then we'll do the whole RNA extraction process, the LAMP process, and that will take around 1.5 hours to 2.5 hours. So the next test is rapid antigen test, and this is something that is going to become more popular. It is much cheaper than the PCR and the LAMP, also quicker. The only thing about this test is that it's not as sensitive as the PCR. Generally, it's only really more useful for those who are actively spreading the virus, who have loads of virus. So it's going to be able to detect those people really well, but those people with a lower amount of virus in their body, it's not going to detect as much. So this is a thing, people are saying that it's going to give more false negatives than it should do, but at least it will be able to quickly and cheaply detect those who are probably the super spreaders, those that have a lot of virus that they're spreading about. So how does rapid antigen test work? This is something you need to know about just in general, the principle of antibodies and antigens. So antigens are specific sequences on proteins that are detected by antibodies. So when your body produces antibodies, your body produces antibodies in response to detecting a particular sequence on a virus or bacteria, and this sequence is known as the antigen. So antigen testing, you'll take your swab, your nose swab, maybe saliva. Saliva is not as good as taking the nose swab, but obviously it's less invasive. You're more likely to have more virus with the nose throat swab. You take the swab, the virus is inactivated in a solution, and also the virus is broken up into proteins, and then there'll be antibodies that are on a strip of paper. You'll put the solution containing the viral proteins onto this paper, and the paper may fluoresce or turn dark or have dark bands if the antibody is able to detect the antigen or able to detect viral antigens. But because this is not like PCR where you're amplifying the genetic material, only if there are loads of viruses that you're going to be able to get this positive result. This can all happen in less than 30 minutes. So again, when we're talking about turnaround time between taking the test or doing the test, taking the swab and getting your results, again, this is really good. But because it's less sensitive, you're not going to get people with lower amounts of virus. You may still be infectious, but just because of the day in which they are, like after being infected, they might not have as much virus. And what people are trying to do is to develop this sort of like pregnancy test, rapid antigen test. So in a sense that you'll be able to take this at home, which will be cool. But again, if you can't rely on this as your sole measure of whether you have the virus or not, just because it's not as sensitive as a PCR. Next, we have antibody tests. So these don't tell you if you're actively infected, but they will tell you if you have been infected before. The thing about antibodies is that in response to a virus, in response to bacteria, your body produces antibodies, but your body doesn't produce antibodies instantaneously. It will take a while. So usually this is a week or more. So this might be like a blood test, a pinprick test. I don't know if they're actually going to draw blood. So the most interesting test that I came across is not actually a biological test, but one that relies on physics principles. So this is the Virolens System. So this one, they say it can give you results within 20 seconds, which of course that speed is phenomenal. So I think they've been trialing this at Heathrow and it sounds really interesting. And again, I haven't read really any research papers about it, but I'm just talking about the technology and how they say it works. So you'll take the mouth swab, you'll put it into the cartridge, which is within their Virolens System, this portable machine. So the machine actually contains a holographic microscope. It's able to generate a holographic image of the surface of your cells. And of course, if there's virus there, it will be able to generate a holographic image of the viral surface. The machine will either tell you whether the sample was positive or negative for the virus. So it's a very interesting technology just to detect quickly, very quickly detect the presence of virus. So of course, testing is going to be a vital strategy to control this pandemic. It might become part of the new normal, might become part of everyday life. Perhaps your business, your company, your organization, whoever you work for, if you're self-employed, you have employees, perhaps you'll be enrolling tests or doing tests just to make sure that you're not spreading it within the business, your company, your office. So thank you for taking your time to listen to this. Please remember to like, comment, share, subscribe, hit that notification bell. And if you're interested in the content that I'm producing, I have other videos which will follow this outro.